z rynku nowych technologii, z różnych branż. Rozmawiamy o nowych rozwiązaniach, dlatego też zachęcam wszystkich o odwiedzenie naszej strony signis.pl/warstwy. Tam znajdą Państwo jeszcze kolejne zbliżające się tematy. Przypominam, że obowiązuje rejestracja na webinary poprzez formularz. Dzisiejsze spotkanie będzie prowadzone w języku angielskim. Po prezentacji zrobimy sesję Q&A, także zachęcam zadawać pytania na czacie. Możecie również zgłosić chęć zabrania głosu, ale też proszę taką chęć zgłosić na czacie, wtedy dopuszczę Państwa do mikrofonu. A teraz przełączymy się na język angielski. So we'll continue in English. Welcome everyone once again in our webinar series Knowledge Has Layers. As I mentioned in Polish, feel free to write your question on the chat during the presentation. At the end, our speaker will answer them, or if you want to ask a question out loud, maybe it will be very specialistic, so you can write down in the chat that you want to take the mic, and I'll give you such possibility. But we will do that in the end of the presentation. And now, please, during the presentation, have your microphones muted. And now to the most important part. It's a pleasure to introduce our guest, Senior Application Scientist, Siobhan. So without further explanation, the floor is yours.
Perfect, thanks for the introduction. Yeah, so I'm Siobhan. I'm one of the application scientists at ONI. So if there's anyone who might not be familiar with our company, we make a product called the Nanoimager, which is the world's first desktop compatible super resolution microscope. So in my talk today, I'm just going to be basically giving an introduction into super resolution microscopy, how it works, and go through some of the biological applications where super resolution microscopy can really help scientists to further their research. So just to sort of briefly outline what the overall mission of the company is. So what we're trying to achieve is we're trying to take these sort of really powerful imaging techniques that are already sort of available, but we're packaging them in a way that's completely unique. And we are doing this in a way so that we can make our microscope more accessible to everyone. And, you know, hopefully if we make our product more accessible, this will help scientists come together and discover more, you know, biological applications and really help them to address their imaging problems. So one of the ways that we make our technology more accessible is that we have multiple different modalities within one single imaging system. So we actually have the capabilities of doing multiple different techniques within the system. So we have the capability of doing dSTORM and palm microscopy. So this structures down to 20 nanometers. Additionally, we have the capabilities of doing live cell imaging and single particle tracking within live cells. So this is a technique that you can actually start to extract dynamic information and behaviors about the molecules that you're tracking within cells. And then lastly, we have single molecule FRET capabilities on the system. So this is really important if you do want to study protein-protein or protein-DNA interactions at a very small scale. So here we're looking at the sort of 1 to 10 nanometer range. The second point that makes the Nanoimager more accessible is the fact that we have different illumination modes within the system. So we can very easily go from epifluorescence mode all the way to TUF illumination mode. And this is all programmed really easily through our software. And you don't have to worry about any laser alignment. Everything is done within production. So you can switch from epifluorescence mode to TUF mode whilst you're actually imaging on the system. So for epifluorescence mode, this is essentially where the light is beaming straight through the sample. So you have a greater depth at which you can image your samples, but as a compromise, you lose some of the signal to noise. If you were then to switch to high-low mode, you reduce the depth that you can image, but then you greatly increase the signal to noise in comparison to epifluorescence. And then lastly, the turf illumination mode. This is quite important if you are looking at any samples that are quite close to the cover slip surface. So this is where sort of the reflection of the beam, as it's totally internally reflected, creates an evanescent wave around 200 nanometers from the cover slip surface. So here you get a massive increase in your signal to noise, but the depth which you are imaging is relatively close to the cover slip surface. So the fact that we can go all the way from epifluorescence to turf, all through the software, makes this a highly accessible technology and quite user-friendly to implement in your imaging. So since I mentioned that the technology is quite accessible in the fact that we can do a wide range of imaging modes and we offer a wide range of techniques, this opens up a range of applications which can be used on the Nanoimager. So it's quite diverse in its sort of research areas. So here I've just chosen to outline sort of 4 applications. And these are key applications that we at ONI actually develop in-house ourselves. So we focus quite a lot on extracellular vesicle research. We look at viruses and bacteria. We focus on neurobiology and also IO applications. But essentially any sort of area where you can fluorescently label a structure of interest will be sort of applicable to at least one of the techniques that we offer on the Nanoimager. So it's quite diverse in a range of different biological applications. So here on this slide, I just wanted to kind of explain a little bit more about what super resolution actually is. So super resolution microscopy is a class of techniques that is able to overcome the diffraction limit of standard light microscopy. So you can actually see that in these images, so here we're looking at B cells in which the BCR receptors have been fluorescently labeled. So you can see here that you get a lot of fluorescent signal, and you can see that there are some structures, but quite a lot of the detail is lost because this is a diffraction limited technique. So if we were then to apply super resolution techniques, you can see a massive improvement in the level of detail that you can actually obtain. So these are the techniques that, in comparison to epifluorescence, you can get a 10-fold resolution improvement. So you can really start to see an increased level of detail of the structures that you want to image. So in general, we always say that by having a greater resolution, this will actually allow us to extract more information, and the more information that you can extract gives you a deeper understanding of your samples and of the sort of structures that you want to image. Overall, super resolution can be a very powerful technique. So just to sort of briefly mention how super resolution works, and why there's a resolution sort of barrier with standard light microscopy techniques. And in order to sort of tell you about this challenge, I'm going to use a schematic of a fluorescent protein here. So typically a fluorescent protein will be around 2.5 nanometers in size. However, when the fluorescence is emitted, the light actually gets diffracted through the objective of the microscope, and it gives you this really large blurry spot. And the size of this sort of fluorescence blur is around 250 nanometers in size. So in reality, when we want to image something that's 2.5 nanometers, we're seeing it as 250 nanometers. So we get this huge expansion of the fluorescent signal. But luckily for us, using super resolution, we actually have the ability to precisely locate the position of that fluorophore. And we're able to do this because light diffracts in a very predictable manner. So if we know the wavelength of the fluorophore and the numerical aperture of the objective, we can actually predict from any one fluorescent molecule what size that diffraction limited spot we expect to see. So this essentially means that in any one imaging system, when we detect this really large sort of blurry fluorescent spot, we can actually work backwards and we can fit this Gaussian distribution. With this fitting, we are able to then detect the precise location of that fluorophore based on the peak from its highest intensity. So in theory, this sounds great, but we all know that in a real biological system, you're never going to get a situation where you will only have one fluorophore and one fluorescent signal in complete isolation. So what is more likely to happen is that you're going to have this sort of high density of fluorophores that are all within close proximity to one another. And that essentially means that all of their fluorescent signal is going to overlap. And this is what gives us the low resolutions within standard light microscopy. So how do we actually then take that system and achieve super resolution imaging on the Nanoimager? So one of the ways that we can actually achieve super resolution is through these dSTORM techniques. So dSTORM is known as a single molecule localization microscopy technique. And essentially how it works is that you have your sample in a special buffer with a dSTORM compatible fluorophore, which you then apply high laser power to exhibit an effect known as blinking. So essentially you'll excite that fluorophore to start blinking. So you should actually be able to see this in the video on the left-hand side. So this is just an example where the actor network has been labeled with phalloidin conjugated to ATO488. And ATO488 is one of these dSTORM compatible dyes. So when we have this sample in a dSTORM buffer, essentially what you're doing is you're exciting the fluorophores to go to a fluorescent on state and to a fluorescent dark state. And this is the behavior that we call blinking. And the blinking will carry on throughout thousands of frames until the final reconstructed image is provided in high resolution. So essentially what this means is that every time we see a flash of fluorescence, we apply that Gaussian fitting from the previous schematic, we find the position of that fluorophore to a high degree of precision, and we localize its position. And we do this over and over again until all of the fluorophores within the image have been imaged, and we do this point by point to get the final reconstructed image. And it's this image that we can get achievable resolutions down to 20 nanometers. So here already you can see from a wide field comparison to a super resolution image, you can achieve a 10-fold resolution improvement. So what this means is that now we can actually start to look at individual molecules, individual proteins, and we can actually start to, at this level, count how many individual molecules there are. So we can start to get this sort of deep quantitative information from the sample. And being able to visualize these proteins and molecules on that level means that now we can actually assess their spatial distribution. And that type of analysis is just not possible if you are using these sort of wide field or diffraction limited techniques. So you really need super resolution techniques in order to get this level of detail. So just to give you a couple of examples of sort of where we've applied super resolution techniques, I've just outlined sort of 3 examples here, and I'll just go through them briefly, sort of one by one. So typically when you use, standard epifluorescence or confocal techniques, you tend to be focused on looking at the cells as a whole. But with super resolution dSTORM, because we're able to hit these really high 20 nanometer resolutions, we can actually zoom right in. So instead of looking at cells just from a whole cell level, we can go one step in and start to look at organelles, and then we can zoom even further and we can start to look at single proteins and single molecules inside the cell or even inside the organelle. So just to point out the first example, so here on the left-hand side, we are actually looking at ion channels on the receptor of mammalian cells. So we're actually looking at two different ion channels, which have been labeled in this example. And these ion channels, they're quite small in size, so they fall below the diffraction limit of standard light microscopy. So we really need the dSTORM techniques in order to be able to actually visualize these small ion channels on the cell membrane. But additionally, through high resolution techniques, we can actually gain an understanding of the interaction of these ion channels. So we can actually see if these ion channels are spatially separated, as you can see here with the isolated just blue and gold signals, but we can actually start to see the ones that are interacting with each other. So you can see that there are some cases where the gold and the blue signals are co-localizing. So it's at this sort of level that you can actually see the single ion channels, but you can also assess their spatial distributions and their co-localizations, which would just not be possible with wide field. The second example is quite a sort of well-researched area, especially within the super-res field, and that is looking at nuclear pore complexes. So these nuclear pore complexes, again, are quite small. So we need these sort of powerful super-resolution techniques in order to visualize single nuclear pore complexes. However, you can actually see in the blue signal here, these nuclear pore complexes are actually formed of 8 subunits. So we, using sort of super-resolution techniques, can actually see the spatial distribution and actually count how many subunits are on the nuclear pore complexes. And again, this level of sort of detail, you really need super-resolution techniques in order to be able to actually characterize and visualize these single nuclear pore complexes. So the last example, I'm actually going to go through in a lot more detail on the upcoming slides, but here we have actually applied super-resolution dSTORM to actually look at virus particles in an infected host cell. And here you can see sort of the whole cell with the virus particles, but you can also see a magnified area where we can actually start to identify virus assembly points, but also start to look at their transport mechanisms at this sort of single molecule level. So with the Nanoimager, we can actually perform super-resolution microscopy in multiple colors. So typically our systems are set up with 4 laser lines, which means that we can actually do 4-color super-resolution imaging, depending on what fluorescent probes you are working with in the sample. So this is a really sort of powerful example of looking at carrying out multicolored super-resolution imaging. And what we're looking at here is actually rat primary cortical neurons that have been labeled for bassoon, HOMA, and V-glue. So what we expect to see is that these markers actually lie on the pre- and post-synaptic side. And we wanted to assess whether we could visualize their spatial distributions within the synapse using super-resolution. And one of the reasons why characterizing the synaptic structure is quite important is because any sort of defects or changes to the proteins within can be linked to sort of many diseases such as Alzheimer's. You can see here, however, that when we actually look at these synapses using just epifluorescence, you can see that there is signal from all 3 markers, and we can see that through the fluorescent blur. but we can't actually identify where they are on the synapse. We can't see how much of the proteins are there, and we can't see their spatial arrangements. So we really need the dSTORM techniques to actually break this diffraction limit and actually look at these markers in high resolution. So you can already see the amount of detail that we're able to get in contrast to the epifluorescence imaging. So now we can actually start to see the formation on both the pre and the postsynaptic side. We're able to actually quantify how much of these proteins are there, look at their spatial distributions, and start to really quantify and characterize the synaptic structure. And additionally, I'll just mention on this slide that the synaptic structure, it is 3D in nature. So we do offer as well a 3D super resolution mode within the Nanoimager as well, which allows us to actually visualize structures in 3D down to resolutions of 50 nanometers. And we can image at depths of plus or minus 0.5 microns on either side of the focal plane. So it allows us to get even more information about your biological sample. So moving on to how super resolution can be applied to extracellular vesicles. So extracellular vesicles is a kind of key area of focus for us at ONI, and it is one where we have sort of created the Nanoimager to be used as sort of a complete tool for EV characterization. So we're able to do this because we can provide the high resolution techniques that allows you to actually characterize single extracellular vesicles, look at their biomarker distributions, assess things like their size, structures, and morphologies. But because we offer live cell imaging and single particle tracking capabilities of the system, we can actually start to look at the dynamic information about extracellular vesicles, such as their internalization, uptake pathways, their biogenesis mechanisms. So all of this essentially allows us to address quite a lot of the sort of open questions that are surrounding the field in relation to EV research. So if we think about how super resolution can be applied to extracellular vesicles, extracellular vesicles are quite small, so they do fall below the diffraction limit of light. So usually we're looking at a small size range of extracellular vesicles that fall between, say, 50 and 350 nanometers. So you will have a lot of extracellular vesicles that fall below the diffraction limit of light, and it is essentially impossible to characterize their surface profile, look at their size, shape, structures, and morphologies through sort of standard wide field techniques. So we really need super resolution to break that diffraction limit to actually allow us to characterize EVs. So just to sort of explain this image here, so what we're looking at, this is a 3-color super resolution image again, where we're actually looking at the EV membrane plus 2 tetraspanning signals. So you can see here that again, on the standard sort of microscope, you can see that there's signal from all 3 markers, but you can't assess where they are located on the EV sort of surface. And, with these sort of dSTORM techniques, this is what we're able to actually characterize extracellular vesicles on a single EV level. So we can actually start to get information on their heterogeneity. We can measure their therapeutic cargo, and we can actually start to profile EVs in terms of diagnostic applications as well. And we have developed quite a lot of the tools needed to actually carry out super resolution imaging in the EV field. So we have actually developed everything from the sample preparation that is needed for super resolution imaging of EVs. We have the technology to carry out super resolution imaging on, thousands of EVs that are bound to a cover slip surface. We have the clustering and co-localization tools that are needed to be applied to the super resolution image to actually extract that wealth of characterization that I mentioned on the size, biomarker, and cargo characterization. So just to give a couple of examples for the EV field. So this is an example where we actually wanted to look at the different populations of extracellular vesicles, and we wanted to see if we could actually look at the positivity. So since we're working on a single EV level, we then wanted to identify, could we characterize how much CD9, CD81, and CD63 are actually present on the extracellular vesicles, and what was the positivity across the whole population. So we actually wanted to get some quantifications to actually say, how many EVs were only single positive for one biomarker, double positive for two biomarkers, or triple positive. And we were able to do that through clustering analysis. So we do provide analysis tools that allows you to extract quantitative information from the super resolution image. And this can also be applied to other biological samples as well. So in our system, we have a large field of view, which means that we can actually analyze thousands of EVs within just one image. So when we perform the clustering analysis, we can get this sort of wealth of quantification. So here is an example where we have actually extracted the positivity of the EVs from the sample. So we've worked out what percentage were single positive, double positive, and triple positive EVs within the sample. And the nice thing about the sort of software tools that we provide to go alongside super resolution, it allows you to get this sort of multi-factor characterization. So we can count and get quantifications for thousands of EVs on the cover slip surface, and we can look at assessing their sizes, their shapes. We can look at differences in morphologies between batches, and we can also characterize their biomarker distributions. Additionally, through these clustering and counting tools, we can actually measure and quantify how much therapeutic cargo is within extracellular vesicles as well, which would be extremely beneficial for any therapeutic or diagnostic application as well. So another way where our clustering tools has been used alongside super resolution dSTORM on the Nanoimager is actually an example from one of our customers where they wanted to look at using super resolution dSTORM in clustering to actually characterize tau proteins, which could be linked to neurodegenerative diseases. So they actually wanted to look at whether this tau protein, which is a microtubule associated protein, was different under a disease or a control state. So what they wanted to see was that could they detect these tau clusters, and could they actually quantify the different states? So could they detect these monomers, the oligomers, the helical filaments, and then these larger sort of aggregated states. And the way they did that was they labeled the tau protein, so you can see that here in cyan, and then they labeled the microtubules, which are in purple. And what they wanted to see was the co-localization of these two signals, and then actually use clustering to quantify how much tau protein was associated with the microtubules. And in this case, the microtubules was just provided as a mask. So essentially, in the control state, what they found was that all of the tau clusters associated with the microtubules, but in the disease state, none of the tau protein aggregates were associated with the microtubules. So they were actually found to be mainly distributed within the cytoplasm, and they were able to quantify this through clustering. Additionally, they use the clustering to actually look at these monomeric states. Excuse me. So if you look at this zoomed in region here, what they could actually see was that they could identify these monomeric states. They could actually identify where it formed these oligomers, and then they could see these large states of aggregation, which are the states which are actually linked to the neurodegenerative diseases such as Alzheimer's. So they were able to actually physically detect the clusters through super-resolution STORM, and applying these clustering techniques, they were able to quantify the size, the spatial distributions, and also use it to look at these different states of disease aggregation. So this was a very interesting way where our customer actually applied, dSTORM and then clustering to their research. And I think this paper is definitely worth reading if people did, want to have a look. So some other areas where super resolution is quite powerful is in virus research. So this is an example here where, we can use these techniques to actually image single virus particles. And these single virus particles, they are quite small, and they will fall below the diffraction limit of light. And traditionally, electron microscopy has actually been used to image virus particles, but EM, suffers sometimes from sample preparation considerations, and you're not able to sort of image multiple different markers simultaneously. So this sort of area is really beneficial for sort of virus research in actually being able to visualize single virus particles through super resolution dSTORM, but actually start to look at, and an even higher level, the spatial distribution of proteins and cofactor hosts within as well. So here we're able to actually visualize a single viral particle, but then actually see that the proteins are localized within a specific region, and we're able to see this visually in high resolution, but again, we're able to extract this sort of quantitative information from the super resolution images as well. So the second part of virus research where we have applied super resolution, and one where we can actually start to see these sort of deeper levels of understanding, is the sort of interaction of viruses within cells and within the cell membrane as well. So this is the example where we're looking at the Ebola-like particle, and we're looking at it in a transfected cell. And what this group actually saw was that through electron microscopy, they saw a sort of difference in intensity when the virus particle was outside of the cell in comparison to when it was sort of in the supernatant. And this is what we could actually see through super resolution dSTORM. So the only difference here is that with electron microscopy, you're working on these really thin sections. But with super resolution dSTORM, we actually have the option to sort of pick and choose the focal plane to actually carry out the high resolution imaging. So what that allowed us to do, which was not possible through EM, was that we could actually start to look at these viruses and these clusters within the cell as well. So we were able to sort of identify these regions here where we had clusters inside the cell, and we wanted to understand, okay, what were these clusters and why, and what were they doing in the cell? So that was the next sort of experiment in this case study, where we had a look at these clusters of viruses within sort of an infected cell and then a control cell, which is just a healthy cell in this case. So you can see here that in the healthy cell, this is a really nice looking cell. You can see the nice cytoskeleton and the actin network. But in the infected cell, you can see here that you start to see these really large clusters, and you can see that the actin network has been visually disorganized, and you start to get these really tight bundled patches. So this group was starting to think, okay, well, do these clusters match up with the actin network in the infected cell? And because we offer the sort of two-color, multicolored super resolution option, we were actually able to see if there was any association. So this is exactly what we did. We looked at the actin network, and then we looked at the sort of Ebola-like virus within the cells. So what you start to see is that where you have these clusters within the cell, you can actually see that they associate to specific regions within the cell. So we identified the assembly sites and then the transport sites. So the transport is where the viral particles will get bundled, they'll get transported, and then this is where they will bud from the cell. So this was sort of the initial phase of just seeing the dSTORM capabilities of the system, and if we could actually use this here to look at, you know, the assembly mechanism, and if we could look at the transport mechanism within the cell. But the nice thing is, because we do have live cell single particle tracking capabilities as well, we could actually do this in a live situation where we look at the live transport mechanisms of viruses. So it does open up sort of a lot of avenues within viral research. So just to kind of briefly summarize, so we do have quite a lot of sort of key features that really do enhance single molecule imaging on the Nanoimager. So I've already mentioned quite a lot through the presentation that we have powerful resolutions. So we can achieve resolutions down to 20 nanometers. And we offer sort of powerful lasers within the system. So in order to excite these fluorophores, in order to get the blinking, we do offer very powerful lasers. We have a sort of autofocus option. So this essentially allows you to find your focal plane within the system and lock its focus. This means that if you move around or if you carry out high frame dSTORM imaging, you won't get any drift within the sample. However, if there is some sort of small drift, you know, the sample hasn't warmed up to the microscope temperature, or there's just any thermal drift from the laser power, we do have a pretty good correlated drift correction within the system as well, which can be applied after the super resolution image has finished. And then lastly, we are compatible with microfluidics. So our systems are compatible with users' own microfluidic devices, but we are developing our own prototype at the moment, which will be fully integrated into the system and controlled through the software as well. So just to quickly move on to the sort of single particle tracking, I wanted to focus most of my talk on the super resolution dSTORM, but because we do offer the live cell single particle tracking, I will just follow up with a couple of examples on those. So this is an example of single particle tracking in 2 colors, and that was to look at investigating the interactions between 2 organelles. So we're actually looking here at the ER and mitochondria contact sites. So our single particle tracking software uses the same algorithm as the localization techniques. So essentially, when the fluorescent molecule is moving, we can determine the position and track where it's moving within the cell to a high degree of sensitivity. So here we're actually looking at PDZ-D8, which is an ER-associated protein, and it's this protein that's involved in ER mitochondria contact sites. And in this example, we're using a halo-tagged approach. And you can see here that the mitochondria has been labeled in green in the background. So you can see here that we can actually look at this system in 2 colors. On the right-hand side, we're looking at the single particle tracks, which are being built up as these particles and proteins are moving within the cell. and we can actually color their output on diffusion coefficient. So you get the speed of these proteins moving within the cell as well. And the really nice thing about this image that I will just highlight here is that as the tracks are building up, you can actually see that they start to look like the structure of the ER, which is, you know, looks quite nice within this image. We can even go sort of, again, one step further with the single particle tracking. So we can actually zoom into the diffusion coefficients and the tracks that have been built up through the software. And we can actually use this to identify proteins that are within close proximity to the mitochondria. So you can see this in this sort of inset here. So we can actually isolate individual tracks for proteins that are moving within close proximity. Equally, you can zoom into tracks which show proteins that have no association with the mitochondria. So this is just an example of a sort of inset that you can get more information from the diffusion coefficients and from the single particle tracking, which is available and you can do in 2 colors as well. So this is an example of single particle tracking to look at extracellular vesicles. So this is an example where the group dosed some cells with extracellular vesicles and left them to internalize for 24 hours. They then wanted to identify if they could identify sort of where the EVs were moving within the cell, how fast they were moving, and if they could get the number of EVs that had actually been internalized. So we were able to actually see that these EVs localized more towards the nucleus, but as they got towards the nucleus, they actually slowed down. So we were able to, through the single particle tracking, actually see that within the cytoplasm, they were moving at a much quicker speed, but as they got closer to the nucleus, they started to slow down. So it could be that they reached their sort of contact point and they localized within that specific region and then slowed down. And all of that we could identify through single particle tracking. And then just to briefly mention for the virus application, so this is a really nice example of single particle tracking capabilities to actually look at the real-time assembly of viruses. So here we're actually looking at a cell that's been infected with the flu. And what you can see through these sort of still screenshots of the single particle tracking software is that you can start to see the difference in where they actually start to assemble within the cell. So you can see that actually in the bottom part of the cell, you can see that there's a lot of particles moving and they're moving extremely fast. So you can see this again through the color code where you can see that the particles are moving extremely fast. In comparison to those moving at the top of the cell, where you can see slow movement, static points. And again, you can see that visually by the short tracks and the fact that the diffusion coefficient is colored in purple. So you can see again that there's a difference in speed and also the track lengths. And all of this can actually be extracted from the single particle tracking software. So for single particle tracking, just to essentially summarize some of the key features before I end my talk, there are a number of sort of key features for live cell single particle tracking. So the first one is that we provide a temperature control. So we can heat the microscope to 37 degrees, which is optimal for any live cell imaging experiments. We have the software algorithms which have been optimized for single molecule detection, and therefore we can track to a high degree of sensitivity using our localization algorithm. We have all of the inbuilt tracking tools within the software. So once you carry out your video for the tracking experiment, you can do all the analysis through the software and get all the outputs on particle number, diffusion coefficients, directionality, track lengths, and everything like that. We can apply the single particle tracking simultaneously in 2 colors. and we do have an extremely high frame rate. So we offer imaging down to 100 frames per second in the full field of view, so we can capture very fast dynamic events. So just to finish off with showing you the actual Nanoimager, so I sort of summarize the applications that the Nanoimager is applicable in. So I just sort of wanted to show you what the Nanoimager looked like in comparison to other super resolution techniques. They're often these very big, room-filling microscopes that take a lot to operate them. You need an optical table, you need supporting infrastructure, and it might take a sort of expert to maintain it if anything were to happen. And our system is not like that at all. It's a tiny black box which doesn't need an optical table or any supporting infrastructure. You don't have to have it in a dark room. It is stable and compatible on any lab bench. And this is what sort of makes us more accessible than sort of other super resolution technologies. And we have done a lot of work in making it very user-friendly, accessible and versatile to make it sort of more desirable to a wide range of researchers. And just to sort of leave you on this side, so it is a desktop compatible microscope. The nice thing about it is it can be remotely accessed anywhere in the world, so you can log onto the microscope and actually carry out your imaging experiments from home, as long as there's obviously someone to put a sample on. And this has been a very good feature for us, especially in current times where, people haven't been allowed into the lab as often as they have wanted to, so having this remote feature is extremely nice. We offer a Python control, so you can implement any scripts onto the microscope as well, and everything can be exported as TIFFs and CSV files, so we make it very accessible in terms of the data output as well, so you can sort of do all of your imaging experiments, all of the analysis for the super resolution dSTORM, single particle tracking and single molecule FRET, but then you have the capabilities of exporting that as well. And then just to sort of summarize that, with the system, you can get a 4 laser line system, which allows you to do 4 colors, which can be very powerful, especially if you do want to do multicolored super resolution imaging. And the last point I will mention is that obviously because of the small size of our system, this does actually mean that it can fit within biosafety cabinets as well. So if you were working in a sort of higher class biosafety lab, this does mean that it can fit into a sort of biosafety cabinet. And we have inbuilt sort of anti-vibration hardware within the microscope body, which means that it is stable under those conditions and in those environments for long periods of time, which is important when you do super resolution dSTORM imaging as well. So yeah, I guess thank you for my talk, and I will hand it over for any questions.
Thank you so much. Thank you so much for sharing your knowledge with us. And please, this is the time for asking questions. If you want to ask it out loud, please write down in the chat that you want to take the mic. And now I will read the question we already have in the chat. So, if this imaging technology is toxic in any way to our samples?
Is toxic, sorry.
Is this imaging technology is toxic in any way to our samples?
So the actual imaging technology is not. So it's a sort of, standard kind of hardware components that wouldn't be toxic to any samples. The dSTORM imaging buffer, if you have a fixed sample, shouldn't be sort of toxic in any way and is compatible with a large range of, biological cell samples, protein samples, DNA samples, bacteria samples. We've not seen, obviously it is a chemical reducing agent, so there is some level of chemical toxic components, but it's not toxic in the sense that it will kill any sample that you want to image.
Thank you. Please, if anyone wants to ask a question, write it down. Or I think if someone will have questions later, maybe you can write it directly to me or directly to Shibon. You saw the email before on the slide. Maybe you can share it one more time.
Yeah.
So everyone can see the email. Thank you. Okay, I don't see any question now, but yeah, remember, feel free to write the email to write with the questions to us. And I think we will finish slowly. So thank you. I want to thank everyone for joining us. Thank you so much, Yvonne, for sharing your knowledge. And I want to welcome everyone to the next webinars that will take place on Friday. Also, feel free to visit our website, signis.pl, and just look what we prepared for you on Friday. Thank you thank you very much to our speaker one more time. Amazing presentation, amazing technology. And one more time, if you will have any questions, feel free to write to us. Thank you so much. Have a nice day and see you on the next webinars.
Thank you.